Hello there, I am Saniya, and today we're going to talk about part 2 of the RNA sequence rescue method in another application of how we would find the absolute counts for the transcripts that we get from our RNA sequencing, and we're going to use the RNA sequence rescue method, and this will be part 2. So please watch the part 1 for a continuation of the application of the problem that we will be working on right now. And please, along the way, please let me know if you have any questions at all. So we're going to be working from the previous part to the current part. We're going to be working on another problem and how we find absolute abundances for transcripts. So I know that we talk about in bioinformatics and biostatistics and in general in health, what we're really concerned with is finding out what goes wrong when we have the disease, for instance. So here, if we've happy SpongeBob, he's like imagination, and then we have sick and tired SpongeBob, I got the suds. And what we're trying to do is trying to find out pathways that are interrupted and genes that are differentially or differently expressed statistically significantly when you go from being healthy to having the disease. And a part of that is, the proteins that we have, so we can go from DNA, DNA has genes, though 2% is, protein coding, for the proteins of the genome. So DNA can be transcribed into RNA, which then messenger RNA, then after processing, it leaves the nucleus and then gets transcribed. So it gets translated by the ribosome into a protein. So we have DNA transcribed to RNA, which is translated into protein. So that's a central dogma of molecular biology. And in a eukaryotic cell, a cell with a nucleus and a membrane, you can see all these different components here. And what we're really trying to do is measure the expression of these different genes inside a cell and trying to figure out what's going on that's different between a healthy SpongeBob and a diseased SpongeBob. Like what genes could be differently expressed, and a part of that is how do we get this data? There's so many challenges. So, as I kind of mentioned in my previous video as well, that we can do RNA sequencing. So we had microarray technology and all these different technologies, and now this is a more modern technology as well. So what you do here is, you have your condition, your tumor, like this is the diseased SpongeBob with the suds, and then you have a normal sample, which is happy SpongeBob, and then you can isolate RNAs, then you can reverse transcribed RNA to complementary DNA, which is called cDNA. You generate the cDNA fragment, size select, add linkers, sequence ends as well. Then you just basically perform high throughput sequencing of the cDNA. And then you can do some downstream analysis of that. So we need some quality control to find and correct for errors with this high throughput sequencing. These mistakes that can happen, even within the cell we have quality control and, DNA mismatch repairs and other things that happen in general. But when we're going from DNA to RNA to protein, we also have quality control. So we should also take and do our part to do quality control when we're trying to measure the amount of, of transcript reads, RNA transcript reads inside a given cell. So one of the solutions is to use RNA Spikens controls. So they help provide and measure sensitivity and specificity of an RNA-Seq experiment. And they're also used in other experiments as well. So this is an example of a paper and as well, and this is like Illumina, which is saying RNA-Seq experimental considerations, each RNA sequencing, experiment type, gene expression profiling, targeted RNA expression, small RNA analysis. It has unique requirements for read length and depth, and some considerations as you're designing this experiment. Like you have to think of some things. And when we get our data, usually like diseased and healthy samples, that we can do differential gene expression analysis, build volcano plots, see the difference in expression, the log 2-fold change, between healthy samples and disease samples, and with the probability of observing that as well. And the volcano plots will tell you the significance, maybe genes whose expression tends to increase in the disease, upregulated, or their expression tends to decrease in the disease. So if this is the control, if their expression significantly increases, they're upregulated, or significantly decreases, they're downregulated. So we can, try to show that in a volcano plot, but essentially what it comes down to, the data I work with, RNA-Seq data, it's important how it's processed. So we need to have like sensitivity and specificity and false discovery rate. Otherwise, we'll, we need to consider the data. Otherwise, we're going to wrongly detect signals when there are no signals, or miss out on potential signals as well. So, there could be artifacts, you remove it, you use filter analysis, trying to, do better, make the results more reproducible. There's a differential expression pipeline that's really important. There could be confounding variables. There's a lot going on. But a key part of what we can get from these experiments is based on the data and how we analyze that data. So here, for instance, this is kind of showing you RNA-Seq data as of now is very modern, current, and very much in demand. So we have indels, which are insertions and deletions, in case you see that here. This is Illumina again. It can detect novel transcripts, single nucleotide variants, indels, small insertions and deletions. Hybridization technology, gene expression is low, and signal saturation. RNA-Seq provides discrete digital sequencing read counts. It can quantify expression changes across a larger dynamic range. So there's so many things, expression dynamics, being able to quantify that information. And you'll see this term, higher specificity and sensitivity. You can detect a higher percentage of differentially expressed genes, especially even if they're low expression as well. You can detect them. That's really cool. You can detect that with this modern technology. Rare transcripts, single transcripts per cell, weakly expressed genes. You can even say maybe if a gene is slightly turned, its expression is turned on a little bit, then you can sort of see and detect that using new technology. So mRNA-Seq specificity is improved, but better detecting transcripts, isoforms, different versions of the same gene than microarrays. It can detect differential expression, increase dynamic range, better able to determine the true genes that are differently expressed between a diseased and healthy individual, because sometimes there could just be noise, and that could really impact. Because if you have like at least 20,000 genes, you definitely know that at least one of them is to blame for some of the diseases that we observe. Most of the time it's it's some genes. And if I tell you, oh, I know all these at least 20,000 genes, let's say I say that all 20,000 of these genes are causing the disease, and you'll be like, yeah, I know that, but I really want to figure out which one. I know that it's in this huge set, but which one is it? It's like an experiment, it's like a mystery. That's what I find so fascinating about this. So you want to find which ones they are, and a key part of that to be able to hone down is to find the special list of genes and to be able to have good data that's reliable. So we were just talking about sensitivity and specificity, and remember this is the healthy SpongeBob versus the diseased SpongeBob. So the source is Wikipedia here, so you can see that this is what we tend to make predictions on. These are the actual positive individuals. So these are the actual positive individuals here. This is the actual positive, and then this is like the actual negative. In reality, we should show it better. This is the actual negative. So out of the actual positive, you're going to have the true positives, which are the individuals who you correctly predicted as being positive. The true positives, TP. That's what TP stands for. You're going to also have your false negatives, which is a mistake. You wrongly predict that this positive patient or this positive individual, positive sample was actually negative. And sometimes, like for coronavirus, COVID-19 prediction, or for instance, imagine you predict somebody isn't infected with COVID-19 when they actually are. So like if you're positive is that they have been infected, and negative is they haven't been infected, have not been. Then a false negative would say that, so if you were to say it in this way, where, oftentimes I think like in healthcare, like the positive is usually like has COVID, given, these are COVID times. And the negative is just saying that no COVID, is that you're not infected with COVID. So sometimes it takes our body a few days to come up with the, like response for an infection or a pathogen or a virus. In this case here, it's a specific type of pathogen, it's a virus. So if you're predicting somebody has COVID, when they actually do, that's a true positive, which is what this is here. So this would be, this class here would be having, this COVID infection. So you actually have COVID. This is the people who have this part here. These are all the people who have COVID. So how many sick people are correct? So usually it's a sickness or something or an infection are correctly predicted as having the condition. That's what sensitivity is going to measure. So, but here, if you positive, you have COVID, a true positive will actually predict you have COVID. A false negative is going to actually predict that you don't have COVID, and that I think is a lot worse, because maybe you don't social distance, or you don't get the help that you need, and maybe you might end up on a ventilator or have like very severe COVID, but maybe you're not showing it right away. So that's, I think, a huge risk. Or if you have cancer and somebody actually a doctor or an AI is telling you that you don't have cancer, but you actually do. They predict that you don't have it, that you're negative, but you actually have cancer, the condition, then you would miss out on a lot of like help and treatment, and your condition can rapidly deteriorate. And maybe you could have been saved, but now you won't. So I often feel like, especially in healthcare and medical applications, false negatives are worse mistakes than false positives. But in a lot of other applications, like sometimes the difference between false positive and false negative is not as bad, right? But false positive might mean that you go through chemo and you spend a lot of money. I know that's important, but like your life could be saved. Maybe you'll have some side effects, but and maybe you didn't have cancer and you went through all this unnecessary hardship. But just think about it, if you had it and you didn't go for treatment, and you could have been saved, but you died instead, or it got so bad. That's why I feel personally that false negatives could be worse than false positives. So here, we also have true positives and true negatives. So true positive is the correct prediction we talked about, and true negative is that you actually don't have COVID. Luckily you don't have COVID. And false positive is we're predicting you have COVID when you actually don't, which is not as bad as predicting you don't have it when you do. I feel that's what I personally feel. And then sensitivity and specificity are just measures of that. So sensitivity is trying to figure out, how many sick people are correctly identified as having the condition. So out of the total individuals who actually have the condition, how many of them are actually predicted correctly as having the condition? The true positives are the correct predicted positive over the total number of positive individuals. The total number of positive individuals is your true positive plus your false negative count. And then specificity is trying to figure out how many negative selected elements are truly negative. So how many healthy people are identified as not having the condition. So out of all the negative individuals, the people who do not have COVID, how many people did we correctly predict as not having COVID? So we're just looking at the accurate results divided by the total number of individuals in that class. So for sensitivity, it's like we're focusing on the positives, and that's why denominator is also out of the total number of positives, how many of the true positives did we find? And specificity is out of the total number of negatives, how many of those did we actually find? And you can see here that, this is the accurate part here, the true negatives and the true positives. So we're looking at the truth. Now, here I'll be talking in this example for the RNA-Seq, rescue method, this RNA Spikens controls. So I just, you might be wondering, RNA Spikens, I've heard about this RNA, virus, and I've heard about like SARS-CoV-2, COVID-19, the virus that sadly we're all dealing with. And you might be like, oh man, is it the same thing? No, and actually this one will be different, though there's a spike protein involved in, SARS-CoV-2, the COVID-19 coronavirus. But no, we're not talking about it here. We're actually talking about something else. So RNA Spikens, what are those? So these are typically RNA transcripts that have two properties. One is that they have a known sequence. So we know that in RNA, it is like A, C, G, or U for uracil. So we come up with a sequence that we want, and then we have like a known quantity that we're going to put inside the cell. And usually, biotech companies and other research institutions, they typically choose a certain solution of RNA Spikens and they use that solution. So it can be used to calibrate and adjust measurements in RNA hybridization assays, DNA, microarray experiments, RT-qPCR, RNA sequencing. Here I also just want to thank Wikipedia. Here's an example, 3D structure of RNA as well. And here we kind of talked a little bit about the disadvantages of DNA microarray experiments, even though I use some of that data in my research as well, but we're kind of moving towards more cutting edge data like RNA sequencing data. So, but you can still use it, but it's a headache to map it like microarrays to genes and do all that analysis, But so since we're focusing on RNA-Seq data, and again, this is a 3D structure of an RNA, so we put the RNA Spikens inside our cells during RNA sequencing experiment. There are many different methods we can use. We can use like bacterial plasmids, other methods to make the cells go from DNA to RNA. We can use RNA polymerase and DNA with the desired sequence to go to RNA. And then what hybridization is measuring. So we're trying to measure, we're putting in, inside our cell, we're putting in some sequence, we're inserting it inside, and a lot of genetic engineering uses plasmids and uses like CRISPR-Cas9, uses a lot of like different ways to insert using these vesicles to insert inside our cells, like certain genes or like, especially like in Drosophila, like the fruit fly or other worms and other experiments and mouse experiments, we can insert all sorts of genes inside them, or upregulate, downregulate, promote, repress expression of certain genes, right? So that's important. In hybridization, what we're doing is we're putting our RNA spike in. It's like a control, like we're putting in a known measurement, of a sequence and a known measure, and these RNA spikins are going to bind to a control probe. So a control probe is a DNA molecule that has a matching sequence. It binds to the RNA spikin, and the process is hybridization. So they just, recognize each other, and then they, like puzzle pieces, they bind. So as I kind of mentioned that, like the hybridization is, like puzzle pieces, like a control probe that we have, the DNA in the cell, and then, this is the RNA spikin. So basically for the RNA spikin, we know the amount of RNA spikin that we put inside the cell. We usually determine that, the solution. Then we see the degree. So we observe, okay, well, what is the actual degree of hybridization between the spikins and the control probe that happens? And then we can use this information here to be able to then say, what are the absolute measurements? So we can then normalize the hybridization measurements of the sample RNA that we have. So it's sort of like a reference point. We talked in the last video on RNA sequence that sometimes we start off using the EM expectation-maximization, where we're looking at the unique mappings from like, a transcript to a read group, the unique ones, using that to get some reference to start with. So similarly, we use a reference. We need a reference, and to do our normalization, figure out the absolute abundances and of the different genes, and to be able to detect that signal, to be able to give ourselves really good RNA-seq data that we can use for our downstream analysis. So we need an RNA spikin, and I often have seen RNA spikins, people talking about them as well, and they're good references. So you're trying to use the spikin, see what happens with the spikin, and then use that information to inform kind of what could be going on with other transcripts and what values those could have, like what's going on in the cell. It's used as almost like a scaling factor. So in the real world, as I was mentioning, you can see the RNA spikin mixes that are available. Like this is an example by Thermo Fisher Scientific. You can buy this here. So what they're saying here is that there's variation in RNA expression data. There can be all sorts of factors like quality of the starting material, level of cellularity, RNA yield, platform employed. There's so many sources. So to control for these sources of variability, so that's what you want to control for these. A common form of external RNA controls has been determined by and developed by External RNA Controls Consortium, so ERCC. If unlabeled, polyadenylated, this is how RNA usually is. It's a poly A tail at the end. Transcripts, they can be added in an RNA analysis experiment after sample isolation, measure against defined performance criteria. What they're trying, they're commercially available. It's what they are trying to do is, you can really unlock the potential of RNA analysis. And as I was telling you here, they are telling you here the exact importance of RNA-seq data. Gene expression profiling, whole transcriptome surveying can lead to better understanding of expression patterns in disease states and provides greater insights into biological pathways and molecular mechanisms that regulate cell fate, development, and disease progression. So traditional methods of RNA analysis like qRT-PCR, microarrays, they're well established, but they're being replaced by next generation sequencing, that high digital alternative. But each method has many platforms, and we need to compare various samples across platforms throughout the world. A standard measure for data comparison is necessary. As the capabilities of RNA analysis expand, it'll be more necessary to create a standardized view of data. So this is all about getting and unlocking the power in your RNA sequencing data. So this is like before you do your experiments, you would have a spike in as well, and there's so many considerations to have in mind. But this is an example of how we use it in the real world. So as I was showing you here, that they talk about how you can unlock it, achieve and compare results. And what they're saying is exactly what I was trying to tell you, that these spike-in controls RNA spike-in controls are being used to create a standard baseline measurement of RNA both within an experiment and across multiple experiments performed using various samples and platforms. So the expression fold change ratio. So I was showing you in a volcano plot, like you could think almost like a volcano, where volcano spewing out this lava, and that's why they call it a volcano plot, where you have like your log2 fold change and your p-value as well. You can like have these log fold change ratios between two samples with a high degree of confidence, when you find your differently expressed genes. So this is like, it's a controls and, flexible options. So it's all about improving the results that you're getting, making sure that you're getting truly meaningful results, such that when you do differential gene expression analysis, comparing sick SpongeBob with healthy SpongeBob and what genes are differently expressed. So that's sort of what really this is all about. So now when I'm going to bring up the question here, so this is the continuation of the bipartite graph from last time with the transcripts and the read groups. So using RNA Spikens to find the absolute abundances for the other RNA transcripts. So here, there are these, different transcripts we talked about in the previous video. They have their transcript length, like transcript 1 is 40 base pairs, transcript 2, 270 base pairs, transcript 3, 1,000 base pairs, transcript 4, 120 base pairs, and transcript X is 100 base pairs. And they all have these different read groups. So let's suppose that transcript A is an RNA Spiken. So that's why you might have been wondering, okay, these others are numbers, and So it's a known quantity that we added in. We added in 1,000 copies of transcript X inside the experimental sample when we were preparing the sample for RNA sequencing. So we got that, from Thermo Fisher or someplace. We just got that solution. We added in 1,000 copies of transcript X. So the absolute abundance of transcript X is 1,000. So our goal is to use the relative abundances of the transcripts that we found, also please see my previous video for more insights, to calculate the absolute abundances. Our goal is to find the absolute abundances for the other 4 transcripts for these transcripts. We want to figure out, okay, well, what did we observe and what are those absolute abundances? So we definitely know going in that we put in 1,000 transcripts of we going in, we know we put in 1,000 copies of transcript X. That's what we went in, with the ERCC spike in. We bought it, we put that into our RNA-seq experiment. So in the previous part, I kind of walked you guys through how we, calculate the relative abundance for the 5 transcripts, here, including the spike in. And how, these are what these values are, is that these were the relative abundances. So we're going to end up using this information on all of these, relative abundances. We're going to end up using it to then find the absolute in this problem. So there's a part 1 to this that I kind of walk you guys through how I did this part. And just to summarize it up, you can see all these numbers, all these characters. This is how we can go about it. So first step is please represent, graph as a read groups, read groups to transcripts. Please then find the reads and transcript lengths for uniquely mapped reads. Please connect unique mapped reads, transcripts, and transcript lengths together. Then find the unique read group for a transcript divided by the transcript length for each transcript. Then you find the unique transcript relative abundance values. Then you update the counts, for each transcript. Then you divide the updated counts by the transcript length for each transcript, and then you can update the relative abundances for each transcript. So it's a very iterative process that's known as expectation-maximization. So again, guys, my name is Saniya. If you found this was helpful, please like my video, and please let me know if you have any questions at all on this first part of the RNA sequence rescue method. Thank you, guys. And in general, you can think of the spiking as like a ruler, as a baseline measure, right? So the absolute abundance of transcript X is equal to the relative abundance of transcript X times the absolute abundance of transcript X divided by the relative abundance. So these values are, this relative abundance of X is the same thing. These are the same things. This part right here, this is the same. These are all the same. So that we found in the previous problem. And what you'll see is that because they're the same, that when we do the math out, if we just cross out that we can just cross this out. And then the absolute abundance of transcript X is just the absolute abundance of X. So kind of going back to this previous slide that we talked about, the spiking controls, part of the RNA extraction, library preparation, sequencing, data evaluation, quantification, input versus output, and the spiking controls are really important there. So what we see is that we know the amount of RNA spiking in transcript X that we put inside the cell. It's 1,000, of that. We also see the degree of hybridization between spikins and control probes, and then we ended up finding between the read groups and the transcript length. So we knew the length that we were putting in of transcript X. We put in a length of that RNA transcript, and we found the relative abundance of transcript X is 0.045379. That's what we determined in the previous video using the RNA sequence method. And then we can use this information to normalize the hybridization of the transcripts of the sample RNA. So again, we knew this information, we observed this relative abundance, and we're going to try to use this information to help normalize and figure out the other transcript absolute abundances. So the key part is for the RNA spiking, of a transcript X, the absolute abundance. So we have the scaling ratio based on the spike in transcript X, where we have the absolute abundance of X divided by this FX rescue. So these are the values we computed, the relative abundances from before. So the relative abundance of transcript X is 0.045379 that we got from here. The absolute abundance of transcript X is 1,000 copies. So now we basically have this information that we were going to essentially use, that the relative abundance of transcript X is, this is the relative abundance that we computed. It's 0.04539, so we got this value from here. The absolute abundance, so we were given this information, and this is what we had kind of put in, before the RNA sequencing data was collected. We kind of went in knowing this would be the, amount we're going to put in. So this is going in as our absolute abundance here. And then we have gone in knowing the relative abundance, which is here. And so this is like our scaling factor that I was mentioning. This is the scaling factor. So we know the absolute abundance, and we know the relative abundance of our spikin, which is here is X. So X is our spikin, our spikin transcript. It's our control. So we just added it in And so that's our spike in our control. So if we want to find the relative abundance for a transcript, we just take the scaling ratio. This thing here is a scaling ratio here. This is the scaling ratio that I was talking about. That's what this thing is here. This is a scaling ratio. This part here is a scaling ratio. We just take it and we're going to multiply it by the relative abundance that we've calculated here. These are the relative abundances. So we multiply relative abundance by the scaling ratio. You know, it's based on our spike of transcript X. So we have the absolute abundance, you know, it's 1,000 copies divided by what we observe relative, our relative abundance here that we observed. So that's what we're going to do. And then we're going to get this. Here's a relative abundance, so these will add to 1, and we're just going to essentially take the scaling factor. So if we have 1,000 of X that we put in, and we have observed, you know, 0.045379, this is our, you know, this is the relative amount of transcript X that we have observed in that cell. So we're going to then say, okay, we had 1,000 that we put in, and this is what we observed. So if this is the total amount and this is what we observed, then this is like the scaling factor, you know, it's just like, you know, rate conversion, slashing and thrashing, dummy, you know, it's that whole like crossing, like stoichiometric calculations where you are slashing and thrashing out the common, you know, doing rate conversions or unit conversions, like, you know, it's like think of it like the idea in a way, if you have like, if I tell you it's something is like 400 and maybe I can tell you something is 485 minutes. How would you convert it to hours, right? So you would say, okay, well, 60 minutes and then 1 hour. And then you can just slash out the unit here and then kind of say, okay, well, this would then be equal to 485 out of 60 hours. So that's like slashing and thrashing. So you can think of it as like almost like that in a way, that if this is this value, this is this value, and this is the total number of transcripts, then if I multiply it by what I observed, and this is the scaling factor, that the scaling ratio that we've gotten from our Spike, and our Spiking is giving us this ratio, that's what we put it in, then I'll figure out, okay, well, what would be the absolute amounts of a given transcript? So that's the absolute abundance. So if this is the total transcripts for X, and this is the relative amount that we had for X out of here, then we just use this as a scale, as a ratio, and then we can multiply our own values by these to find their absolute values. So it's like a ratio. This is absolute, 1,000 is absolute, is to relative, and this is a relative here, so we can find the absolute for that transcript. I used to love like those types of problems. So again, like this is the same example here, where you know, we just put in these values and we find what it is. So please note the absolute abundance for a transcript therefore is going to be equal to this, you know, like relative abundance times the scaling factor, 1,000 transcripts of our Spiken divided by this relative abundance of our Spiken. That's what this is, our relative abundance of our Spiken. And that's how we get the absolute abundance for a transcript number, you know, for a transcript number. Oh, wait. So they kind of again what I'd shown earlier. So if we want to find it for transcript X, which is our RNA Spiken, we just take this here, this value here, and we're going to plug it in here for this rescue. We're going to apply that formula here, put this into our rescue, which is this value here, and we have the same scaling factor here that we had determined. And then you're going to notice, hey, we can just actually cross out this value and this value, they cross out, so we're left with 1,000, which is what we expected. Absolute abundance for transcript X is 1,000, right? That's what we put in. So that just confirms it's the same thing, right? You get the same result that you expect. So the formula is great. So similarly, like now we can use the same formula, and since it's transcript 1 that we're looking at, you know, transcript 1 here, that's why we use f of 1 here. So we're going to just take this value here, take the relative abundance for transcript 1, and then just put it in here. And that's why we get this value here. Oh wait, it should be here and then here. And the scaling factor that I was talking about, this value here, the scaling factor, it actually, if once you put in the values, it actually evaluates to being 22,036. So that's like the scaling factor. So you're just going to multiply your relative abundances by this 22,036. That's like a scaling factor. We just multiply them by that, and then we'll go to our absolute abundances from here. So once we take this value, you know, this relative abundance value, and then we multiply it by the scaling factor that we got from our Spiken, we then get the absolute abundance for transcript 1 is 12,609.09. So we can do the same thing for transcript 2 as well. We just have to take this this relative abundance for transcript 2. We just have to take this value here, and then we just have to plug it in here. And the scaling factor, everything else, this part, all the same. We just multiply. So essentially the way that it goes is you just have to take the, to figure this part out, all it is the absolute for transcript number. I'll just write T number is equal to relative. So it'll just be the relative for the transcript number times the scaling factor. And then it'll just be spike scale. So again, as I was just mentioning, you know, like we can just find the absolute abundance for transcript number. It's, you know, just equal to the relative abundance for that transcript number, which, you know, we're talking about the relative abundance, you know, here, multiplied by the Spiken scaling here. That's what, you know, this value is here, which is what these values are. So you just have to multiply that Spiken scaling, that ratio that we get, you know, taking into consideration, you know, the Spiken control, which is X, and then the absolute number of transcripts, the absolute abundance of the Spiken divided by its relative abundance. We use it as a scaling factor, multiply it by the relative abundance of the transcript of interest, which is here, which is 0.116121. And then we'll get our absolute abundance for transcript 2. And then the same thing for transcript 3 again. Just take this value here, this value, and then we can multiply it by our scaling factor, and then we will get the absolute abundance, you know, so absolute, the same scaling factor, the absolute abundance for transcript 3 will be 4,820.512. And last but not least, the same idea for transcript 4. You know, we focus on transcript 4, the absolute abundance. So we take the relative abundance for transcript 4, we multiply it by our scaling factor, and then we'll get that the absolute abundance for the RNA transcript 4 is 1,047.842. So that's kind of showing you in a way that RNA spikings are like a benchmark, you know, kind of connecting the puzzle pieces. They're like a reference point, you know, like when somebody says, oh, I love you from to the moon and back, right? It's like a reference, it's a benchmark, right? Of course, they love you way more than that, right? Hopefully, like a mama bear and a little child bear, right? It's like a reference point, and that's what RNA spikings are. They're like a reference point, right? It's all relative, right? But now the people are trying to use distance to measure love, I guess. They can add arms, an arm, heart, I don't know, like, you know. So we're trying to use a benchmark, right? People often say that the size of your heart's like, you know, a fist size or something, you know, it's a benchmark. So then RNA spiking is like a benchmark. It's a control we put in so that we can try to go from these relative counts that we observe to more absolute measures to say, okay, we put in this amount and we knew that we put in this amount in our cell. We definitely knew that. It's something we know going in. And then we observe this. So we observed these relative values, and we knew that this was our absolute values. So can we try to use this as a proxy, as a way to try to estimate and normalize what could be the absolute counts of the gene in a cell that we get? So it's like a proxy to try to help us figure out, you know, what's going on inside the cell. And, you know, over time, these technologies are going to get better and better. But for now, that's sort of like a way to get more reliable RNA-seq estimates, get better data, and then we can use that data to get more reliable results and better help people. So in the end, I just want you to see that, we can find these RNA absolute abundances. And we knew going in that we would have 1,000 RNA sequences for, for our spike in control X. We knew this. We designed the sequence as well that we put in. We designed the base pairs, and we, knew all of that going in. And then what we found is we saw the hybridization results, like the pairing that happened, and we observed these relative abundances. This is again from the previous video, so please let me know if you have any questions on that. So that was part 1. We found these relative abundances. So we could use the relative abundances and use these 2 parts to define our scaling factor. So again, we could use these 2 parts, define our scaling factor as just being 1,000, this absolute abundance over this, let me see, put this 0.045379. So we knew this value, and then all we have to do is just multiply our results by multiply for a given for a given transcript. Just take its relative abundance, multiply it by the scaling factor here, and then from that get a value, get our absolute abundance. That's all you have to do. Just take our so I'll explain again. So we knew going in that we put in 1,000 transcripts of X. We knew this ahead of time. We found from the previous part, from our bipartite graph, we figured out these relative abundances. So we found that the relative abundance of transcript X, again these add up to 1, the relative abundance is 0.045379. And then what we did is we took our absolute abundance divided by our relative abundance, used it as a scaling factor. So this is like 22,000, I think 300 something when you do the division. We have our scaling factor, and all we're doing is just multiplying our relative abundance, it says value for a transcript, by the scaling factor to then get our absolute abundances. So these are the absolute abundances inside the cells that we found. So about like 12,609 of transcript 1, about 2,558.915 of transcript 2, 4820 of transcript 3, 1,047 of transcript 4, which is very close to what we find for transcript X. So it's, that's just to try to tell you, how we did this and how we can go from relative to absolute abundances, all by using our RNA Spiken. Yes, we did it. So thank you guys for watching this video. If you have any questions at all, please let me know, and I'll be happy to help and explain the concepts. Thanks, guys. And please like my videos or write any comments if you have any. Thank you.